যত সব বিয়ে করছো তুমি, তুমি নিজের ফ্যান্টাসিস পুরো করার চেষ্টা করছো আর কিছু। এটা করা অনেক আমি তো শুয়ে শুয়ে কাজ করে দিই। এই কাগজ তো এখানে মহানদীর পাশাপাশি বসে একটা অফিস করবে, ওদের একটা ঘর আছে, এদের বিশাল করে এদের ওই নিজেদের ওরা কাজ করে এই রুমটাতে বসে। আমি বললাম না দেখে এগুলো অতটা হবে না, অন্য কোনো জায়গায় পাওয়া মুশকিল, সবাই এখন ঘুমানোর জন্য ব্যবস্থা করছে। চুচুড়ি চুচুড়ি মাছের ডিমের বড়া অসুবিধা দিয়ে যা রান্না হয়েছে খেতে হয়ে যাচ্ছে। বলছি যে ঘুমাতে চলে গিয়ে কালকে একটা কমপ্লিট অর্ডার করে দিয়ে খাওয়া হচ্ছে। চারটে বাজতে চলেছে, আমি রেডি হই, ঠিক আছে? লোকটা দেখো ভালো কিছু। ওকে আগে ওখানটা করিয়ে নিতে বলো, মাঝখানে একটা ব্রেক নেওয়াটা ইজি হবে। ওকে আগে ওখানটা পুরোটা করিয়ে নিতে বলো, তারপরে মাঝখানে ব্রেক নেওয়াটা ইজি হবে। প্রথম প্রথমে ঢুকতে দিও না এই রুমে। হ্যাঁ, সেটাই বলো। So hello everyone, let us get started for the day. Okay, welcome everyone to the week 9 PMRF-NPTEL session on molecular biology. Today we are going to study a lot about PCRs and their use in modern molecular biology to understand different things. Yeah, yeah, yeah. Okay. So, a brief introduction from my side. My name is Anuvab Chatterjee. I'm a PMRF fellow at IIT Kanpur, and I will be taking this PMRF-NPTEL session. As you would know, these sessions are designed so that uh, the people that are taking the NPTEL courses have a live session with someone who is taking the course, such as myself, and we discuss various concepts behind the questions and quizzes that are posed during the NPTEL course to and solve them together to get a better understanding of how exactly to tackle the questions as well as understand the concepts behind them. So, as you would know, how we do this session is that I will show you a question from the previous year's run of the course, and I will give you some time to figure the answer out, and then together we will try and solve the question. So, for today, as I said, we are going to learn a lot about polymerase chain reaction or PCR and their use in modern molecular biology. And let us get started. So, the first question for the day is what is the function of SSB protein? The options are they prevent re-annealing of single strands during DNA replication, they increase DNA polymerase activity, they prevent DNA degradation, and lastly, they assist in primer synthesis. I will give you a minute to figure this one out. Do এই রেকর্ডিং আরম্ভ করে দিয়েছি ধন্যবাদ ধন্যবাদ Okay, well, that should be enough time to try and answer this yourself. Let us get down to it. We have been learning a lot about DNA replication in the central dogma, and one of the PMRF-NPTEL sessions of the past weeks was completely dedicated to it when we were learning about various basic fundamentals of molecular biology. And we are revisiting that in order to get to how exactly we use PCRs to do DNA synthesis. So, just a basic uh, reminder for how DNA replication works. DNA replication is the process in which the cell by itself replicates its DNA. So it could be the entire genomic DNA that is getting replicated or the plasmids that are getting replicated. So that you have two copies of the DNA that then get separated equally amongst two daughter cells. So there are three different steps to DNA replication. The first is initiation, which prepares the DNA for synthesis. In this, we actually have all the different proteins that are used to synthesize the new strand of DNA, bind to the DNA. This you would remember being the helicases, the topoisomerases, the gyrases, the SSBs, the different primases, and most importantly, the DNA polymerase, that is a DNA dependent DNA polymerase that will bind to the strand, actually read the DNA sequence and synthesize the complementary strand of the DNA, and then opening of the double helix of the DNA. Next is elongation or synthesis phase, in which proteins connect to the correct sequence of the nucleotides into a continuous new DNA strand. In this particular step, actual DNA synthesis takes place, and now the DNA replication is walking along the template DNA strand and synthesizing the corresponding complementary strand using DNTPs that are floating around. And finally, we have also discussed termination, which is the stopping of the DNA synthesis, in which the proteins that were bound to the DNA are released. This can happen either in a row dependent or a row independent fashion and depends on the replication termination signal. So this all of this so that was for transcription. For replication, there is the termination works a bit differently, but this is essentially the three different steps for DNA replication. And we have already seen the different proteins that are involved in this. It includes the helicases that separate the two strands, primases that synthesize the RNA primer on which the DNA polymerase will sit and start the synthesis process, and SSBs or single strand binding proteins. So once the helicases actually separate the two strands and form the replication bubble so that the DNA polymerase can sit on the single strand and start the synthesis. In order to prevent the already separated two strand to come together and rebind into rehybridize into a double helix, there are SSB proteins, single strand binding proteins. These proteins can bind to single stranded DNA and stabilize it so that it doesn't go on and again rehybridize, which will cause a lot of energy to be given to keep the strands open continuously because the helicase has to go over the same strand again and again. So SSBs do a very important job of keeping the replication bubble open so that DNA synthesis can take place. Then we have DNA polymerase, which synthesizes the new strand of DNA, and tethering proteins, which stabilizes the polymerase. So, tethering proteins bound essentially make sure that the entire complex stays situated very well on the single stranded DNA. So, this is how DNA replication works. So, way back when, around the 1960s ish, we actually, when we wanted to synthesize DNA or replicate DNA in vitro. So you say have a piece of genomic DNA or a gene in a plasmid and you wanted to replicate more copies of it. That was a very tedious task because the only way for us to do it is use all of these different enzymes at the same time in the same single pot, which was known as a single pot reaction, in which you actually lyse the cell and then put in everything that you require for the synthesis to take place, and then use some a polymerase such as the so you use the DNA pol 3 in order to actually do the replication. And it was a very tedious and difficult process because at once the DNA polymerase could only synthesize one copy of DNA, and it took a lot of time to actually synthesize a lot of copies. And the entire process was pretty tedious. But all of this changed when we actually standardize the PCR or the polymerase chain reaction. We'll see how that works. But essentially, there are three different processes, as you would have understood from the entire DNA replication part, that there are three main steps that need to be done in order for DNA replication or DNA synthesis to take place in vitro as well. The first is initiation, in which you prepare the DNA for synthesis. In this, the entire idea is you just separate the two strands out. Normally, in vivo, this is done by helicase, which breaks the hydrogen bonds between the nucleotides at the cost of ATP and keeps separates the two strands apart. But we figured this out that if you heat DNA at 95 degrees Celsius, which is way, way higher than its melting temperature, then the strands will separate just on the basis of the heat that you have provided. The kinetic energy of the molecules increases, the hydrogen bonds break, and they separate. So you can replace the helicase enzymes with just heat. The second is a synthesis phase. In synthesis phase, we would require a polymerase that can do it. And there were various different polymerases that could have been used, but the major breakthrough came when we figured out that with the discovery of TAC polymerase or thermus aquaticus polymerase that can actually work at a very high temperature, which is around 72 degrees Celsius. So the optimum temperature for synthesis of TAC polymerase is 72 degrees Celsius, not the normal 37 that we would expect. And also it is heat resistant. So you can heat the entire polymerase up to a very high temperature and still it would not be nature. So, just a moment. Yeah. So you can heat the tacpole to a very high temperature, such as 95 degrees Celsius, and even though it will stop its normal function when you cool it down to 72 degrees, it will restart polymerizing DNA. This is a very important step because otherwise you can only use the enzyme for one time. When it is heated up to 95 degrees, the temperature that is required for the DNA to denature, you and you put in the enzyme, the enzyme will also denature and will not work. So this actually gives us a very strong tool, a powerful tool to keep on synthesizing DNA again and again. How is that? Say you have denatured the DNA using heat at 95 degrees Celsius. So if you let it cool down to 37 for normal DNA polymerases such as the Kornberg enzyme or DNA pol 3 to work, so the DNA will again renature, and now renatured DNA, double-stranded DNA polymerase will not be able to work on it. So you would need a polymerase that works at a very high temperature, and that polymerase is TACPol. So once you put in Taq polymerase at 72 degrees Celsius, the DNA is still has not been able to re-anneal and form a double-stranded structure. So our DNA polymerase keeps on working. And the final step that we have seen during DNA replication also is termination of DNA synthesis. And this can be also done very easily by lowering the temperature. Once you lower the temperature, the Taq polymerase becomes inactive. So you normally lower it to 4 degrees or 16 degrees, and that will stop the reaction. Then you can even go ahead and try to heat inactivate the enzyme or purify the entire DNA that you have taken out, and essentially you have synthesized your DNA double strands. The process of PCR or polymerase chain reaction actually does all of these three things in a cyclic manner so that you get exponentially larger amount of DNA from a very, very small amount of DNA, the starting DNA. We will see how that works in a bit. So the function of the SSB proteins or the single strand binding protein is it prevents the re-annealing of single strands during DNA replication. Next, the next question is, the whole process of polymerase chain reaction involves three main events. What are the three events? The denaturation, transcription, and translation, denaturation, annealing and elongation, denaturation, transcription and elongation, and initiation, elongation, and termination. So I would let you think for a bit and try and figure this out. Jagannath says it is B, denaturation, annealing, and elongation. Okay, so let us look at it. So as I was saying, we are going to discuss PCR, and here we are. So PCR is called polymerase chain reaction. So if you divide the names into three parts, polymerase because you are obviously using a DNA polymerase to synthesize new strands of DNA, and it is a chain reaction. A chain reaction is something that you initiate and it keeps on growing exponentially, and we'll see how that works. So PCR is a repeated cycle reaction that involves mechanism of DNA replication. But instead of using the 15 different enzymes that you are required for DNA replication in vivo, you replace most of them and then only use the bare minimum, which is the DNA polymerase, which in this case is the heat resistant Taq polymerase. So PCR results in the production of multiple copies of DNA from a single one, and the whole process is divided into three different parts: denaturation, annealing, and elongation. So the denaturation and annealing is the initiation part, like simulates the initiation parts of DNA replication, and then elongation is just elongation. And instead of a termination, particular termination sequence or a termination signal being there, you just lower the temperature from the optimum enzymatic DNA synthesis temperature of 72 degrees Celsius, and that just stops the reaction. So a DNA fragment of interest is used as a template from which a pair of primers or short oligonucleotide complementary to the both double strands of the DNA are made to prime the DNA synthesis, where the direction of synthesis or extension is all 5' to 3', as in DNA replication. So DNA replication still often happens in the 5' to 3' direction, and the number of amplified DNA or the amplicons increases exponentially per cycle, Thus, one strand, double strand of DNA gives rise to two double strands, and then 2 will give rise to 4, 4 will give rise to 8, and 8 will give rise to 16. So, an exponential increase in the amount of DNA that you started with. This is described here as well, and also in the next image that we'll look at. So you start with a single strand of DNA and you double it, and that two single strands of DNA can act independently as templates for the next round of synthesis. So now you have two single strands, and then you can synthesize the complementary strands for both of them, and then you end up with four. And with the again with four, you now have four separate templates, and you again do another round of polymerization to find eight different strands. And this can continue as long as you keep on giving the reaction DNTPs or ATP, as well as the polymerase keeps on working. And thus it is a chain reaction. It increases exponentially with a very minimum input. So how does polymerase chain reaction work? What are the requirements? The first requirement is a DNA template that you would want to amplify. And then you have primers. As you would remember from the DNA replication lectures, that DNA polymerase cannot sit on just single stranded DNA. It requires an initial primer or a short oligonucleotide sequence that is already bound to the single stranded template that you want to amplify. So, our DNA polymerase sits on both the primer and the DNA template and adds new nucleotides to the primer. So, without the primers, it should not work. So even in PCR, the same thing happens. You need primers, and these are, instead of being RNA primers that we see in replication, we provide DNA primers for PCRs, and these will bind to both the leading and the lagging strands, or rather, not the leading and the lagging strands, in the top and the bottom strand, and then the entire synthesize the entire new complementary strand. Next, you obviously need the nucleotides, which will actually be polymerized. You need Taq polymerase, which is the heat-resistant polymerase that we use, and the PCR buffer that provides all the ample conditions for PCR to take place. So now let us go through the different steps. And the first step is denaturation. As you would remember, you need helicase to pull the two double strands apart. But when you apply heat, that alone can cause the same effect. It pulls the two strands apart and denatures the double-stranded DNA into single strands. And essentially, if you keep the temperature high, then the two strands will remain separate. So denaturation occurs at a very high temperature of 95 degrees Celsius. Next, you need to anneal the primers. So the primers will go and bind. For this, you lower the temperature to 55 degrees Celsius, and this allows the primers to go in between the two strands and bind. And here also there is this idea that if you lower the temperature too much, then the double strands will start coming together and binding again. The only reason primers can bind to the double strand and not the two huge strands binding together again is first the small, since the primers are very small, these are 18 to 24 nucleotides long. So denature re-annealing of primers to the template requires is much faster because they only need to anneal just a small part. And secondly, you put in a huge amount of primers in comparison to the initial template that you add. So it essentially is a competitive reaction in which the annealing of the primers is actually much faster kinetically, and thus they go and just bind much faster before the two bigger strands can anneal together. And finally, extension. You then increase the temperature back to 72 degrees Celsius when our Taq polymerase can bind to the primer as well as the template strand and start polymerizing the complementary strand. And hence, you have essentially in one round of PCR from two, so one single strand, one double strand of DNA made two double strands of DNA. And once you keep on repeating this cycle of denaturation, annealing, and extension, you will get exponentially more amount of DNA. So from one you made, so essentially from one double strand you made two. Next round, this product, which is one of the older strand and a newly synthesized strand, will serve again as the template, and thus from two double strands, you will be able to make four, from 4 to 8, to 8 to 16, and you will be essentially able to make a huge amount of DNA at the end of the polymerase chain reaction. And here we have a bit more detail about the different steps. So this is how what the polymerase chain reaction that I described looks like. This is the stage two. First, you denature at a high temperature of 95 degrees Celsius. Then you lower the temperature to allow for annealing. This is around 55 degrees Celsius. And then you increase the temperature again to 72 degrees Celsius to allow for Taq polymerase to synthesize the new strand of DNA. And this thing repeats again and again and again. And this is what you say is the number of cycles the PCR has been running for. You always aim for more than 30 cycles, which actually is huge amount of DNA that it will render you. And as you would imagine, the last later steps of the PCR actually give you much, much more DNA than the initial steps. So now, what do I mean by that? So initially you are making 2 to 4, 4 to 8, 8 to 16. But in the last steps, you are making 1000 to 2000, 2000 to 4000, 4000 to 8000. So essentially at the last ending of the PCR are the most important steps in which we actually exponentially amplify the entirety of the DNA. There are two more ideas here. First, so denaturation you can do for 30 seconds, which will almost always denature your DNA. But if you have a very big DNA, then you can increase this time. The annealing temperature actually depends on the length and the sequence of the primers itself, and we will see how that works. And finally, the extension time, how much longer the PCR will run at 72 degrees Celsius. This actually is determined by how much of the template you are trying to amplify. So if you say if you are the part that you want to amplify is only 1 kb, so Taq polymerase as uh, like we say that Taq polymerase has a processivity of 1 kb per minute. So it makes 1 kilobase pair extension per minute. So say that your DNA was around 1 kb in size, you will put the extension time as 1 minute. When you put it at 2.3 minutes, which is 2 minutes and 30 seconds, you imagine it will be able to amplify 2.5 kbs of DNA. So this all depends on how long your DNA is and how much you want to amplify it. There are two more steps, which is called the initial denaturation and the final extension. So the initial denaturation is done at the similarly high temperature of 95, and you can do it from 5 to 10 minutes. And this step is important so that even before the PCR cycle can start, you absolutely completely denature everything. So it should not be that you are only using 30 seconds to denature the template, but you use entire 5 to 10 minutes to make sure everything is denatured before you start the first reaction. Next is the denaturation that is normally part of the cycle, and this is the first step in which the double-stranded DNA is denatured to two single strands. Next is annealing. This is the annealing step where at a lower temperature, usually about 55 degrees Celsius, but it depends on the primer sequence. Primers are allowed to bind to the template DNA, and the annealing time is 15 to 30 seconds, depending on the length and base of the primers. Then C is the elongation, and this is the synthesis step where the polymerase performs the synthesis of the new strand in the 5' to 3' direction using primer and the DNTPs. Average DNA polymerase at 1000 base pair per minute, and this entire 1, 2, 3 cycle is repeated from 30 to 45, 30 to 40 times for typical amplification. And if you do the math, then you will see that after 40, it doesn't matter if you're running the PCR because that will give you so much more DNA that you probably will run out of DNTPs by the time. So around 40, 35 to 39 cycles is on average how long a normal PCR lasts. Then after the cycles are completed, the reaction is allowed held at the final denaturation of 72 degrees for 10 minutes and will allow for any strand which was not completely synthesized to complete its synthesis. And then you finally hold the PCR at 4 degrees to stop the reaction and also prevent any more any damage to the DNA because of heat or anything. So this is essentially how the entire PCR works. So here is again how the exponential form of the PCR works. You have the original DNA to be replicated. You denature into the two strands, top and bottom. You anneal your primers, and then you allow it to elongate. Once you have elongated, you essentially have this particular newly synthesized double strand, which is exactly identical to the original, just with the addition of the primers. Now, this double strand can again take part in the same reaction. You then denature it, then anneal new primers, then elongate, and this entire process can keep on happening again and again and again. So essentially, from one single strand, you have made two single strands, one double strand, you have made two double strands of DNA. Then you repeat it again, you can make four double strands. Repeat it again, you can make eight double strands. So exponential increase in the amount of DNA you have. And these are the different times that we have already discussed. Initial denaturation is 95 degrees for 5 minutes. Uh, Denaturation is 95 degrees for 30 seconds. Annealing is 55 degrees for 30 seconds. Extension is 72 degrees depending on how much you want to extend. And final extension is 72 degrees for 5 minutes. And holding is 4 degrees to stop the entire reaction. And this entire process, you don't have to manually. So it was that originally that people used three different water baths for this. One water bath was set at 95 degrees, one was set at 55 degrees, one was set at 72 degrees, and you individually took the tube from one to the next, and then repeated it again and again. So this was a very manual process, and we were able to extend DNA that way, but that was tedious as well, prone to mistakes. And now we have something known as a thermal cycler. This is this box kind of a machine that does all of this at the same time. You just close the box, set the program, and it will go through the cycles by itself. So now we have automated the entire thing, and the PCR is now as basic as having a pipette in your lab. So the three steps for polymerase chain reactions are denaturation, annealing, and elongation. Any questions? Okay, moving on. The next. During PCR, the annealing time of primers to the DNA strand depend on what? The pH of the solution, the length and basis of the primers, the temperature of the reaction, or the concentration of the DNA. I'll give you a minute to figure this one out. How much is it? Is B, length and basis of the primers. Jagannath also says it is B, length and basis of the primers. So let us look at it. So what are the primers for a PCR? We have already seen it. Let us go through it again. A primer is this short DNA stretch that serves as the starting point for DNA synthesis. In DNA replication in vivo, this primer is synthesized by RNA primases, but in case of PCRs, we actually provide the primers by ourselves. This actually allows us to select which part of the DNA gets amplified. For example, if you have the entire genomic DNA, which is 20,000 base pair long, and you only want to amplify a 1,000 base pair region or 500 base pair region, you don't have to amplify the entire thing. You can just add two primers that are flanking the region that you want to amplify, and that will just amplify that 500 base pair of this 1 kb region that you want to. So primers are really important part of PCR. And in PCR, two primers are required to bind to each of the single-stranded DNA, flanking the target sequence, and these are known as forward and reverse primer. The forward primer binds to the top strand, reverse primer binds to the bottom strand, and amplifies. But always the amplification happens in the 5' to 3' direction. They these primers have sequence complementary to the sequence so that can sequence of the target so that they can bind and start synthesis. And how exactly do you design these primers? So nowadays, it used to be tedious, where you actually figured out a lot of parameters by hand. Nowadays, there are multiple websites or applications that you can use. The one that is still relevant that we use is known as Primer version 3, just Primer V3. or you there is primers can also be synthesized using the primer blast. So if you have ever gone to the blast page on NCBI, then you can just look for primer blast and give your sequence that you want to PCR, and then it will just give you the primers that are required. So there are some considerations that are required for primer synthesis. The first is that they are 18 to 24 bases, which is the ideal length for specificity and short enough for primers to easily bind to the temperature template at the annealing temperature. So as I said, primers need to be shorter than like way shorter, because you need them to quickly bind to the target strand before the target strands could bind with each other, because the target strands are also DNA, and at 55 they will also try to bind together, but you need small primers so that they quickly go and bind, and you put in a lot of primers so that they competitively outcompete the two target strands and can bind. The second is specificity. The worst that can happen during a PCR is that your primers binds to somewhere you do not want to. And now when you run the PCR, you get a different band than the one that you wanted because your primers went and bound somewhere else. And thus you have to find a sweet spot between specificity and the length of the primer, and that is around 18 to 24 nucleotides. And whenever you design PCR primers for amplifying something from, say, genomic DNA, you should always make sure that you run a primer blast. The primer blast, what it will do is that look at all other sequences that the primer can bind to, and you should always be aware that your primer can bind to some. If your primer binds to somewhere else, you will non-specifically uh, PCR amplify a lot of other things that you do not want to. So running primer blast is an important step and a general procedure nowadays. You make sure that your primer binds to where you want and not where you do not want, and give you unrelated bands. Next is the primary melting temperature. So as you would know, melting temperature is the temperature at which the DNA strands can separate or denature from each other. And primer melting temperature needs to be around 52 to 58 degrees Celsius to give you the best results, because you want it to anneal at 55 degrees Celsius or close by. So how do you find out the primer melting temperature? There is a very easy calculation that you can always do, which is 4 times the number of G's and C's and 2 times the number of A's and T's. This gives you approximately the amount, the melting temperature at which your primer will melt. And for very big amount or very long primers, you can use this complicated process, but this almost always gives you a very good approximation. And if you're using a software such as Primer V3 or Primer Blast, they all already automatically calculate. So this is a better formula to remember to calculate the melting temperature from the back of your hand. And finally, so melting temperature is not what you want. What you want is the annealing temperature, the primers, where they can bind to the double-stranded DNA. And but in order to calculate that, you will have to calculate the melting temperature, because annealing temperatures is around -5 degrees from the melting temperature. So there is again a calculation which gives you the correct annealing temperature. This is empirically found, so people tried again and again to figure out what the formula is, but it is around 5 degrees from the melting temperature. So you just figure out the melting temperature, and then you figure out the annealing temperature. And if the annealing temperature is too high, then it will lead to insufficient hybridization, because if there is too much heat, then there is too much kinetic energy, and the primers will not bind or anneal properly, and it will give you very low results. And a very low annealing temperature will cause the target template strands to also bind together. So that will give you a higher number of mismatches, and also it will reduce the stringency of where your primers can go and bind. So at 45 degrees, the primers can go and bind to a lot of different places, because some bases will always hybridize to somewhere, and you have 18 to 24 bases in your primer, so they might bind somewhere non-specifically and give you a lot of bands. So keeping it around 55 degrees is around the best way you can do it. So that is how you design a primer. You figure out the length of the primer. You figure out what sequence it should flank. You figure out that it is non-specific. You figure out the melting temperature that gives you the annealing temperature, which should be around 55. Then you're good to go. And one other thing that you should keep in mind is that when you design primers, you design a pair of primers. The forward primer that will synthesize the top strand, the bottom primer, the reverse primer that will synthesize the bottom strand. Or I'm saying the opposite way around, but yeah, yes. So when you design the two primers, you're designing two at the same time. So they logically they should anneal at the same temperature. So if one of your primers anneals at 55 degrees, the other anneals at 60 degrees, but you have to then set your what will you set as your annealing temperature? And so that will give you a lot of problems because then one primer will bind, the other will not. And as you would have seen from the PCR schematic, you get exponential amplification only when both the primers bind. So you have to make sure that both of the primers are around 2 degrees from each other at annealing temperature. Otherwise, one will bind, the other will not. You will get not get an exponential increase in DNA amount, which is why you are doing the PCR. So these are all the points that you have to keep in mind when you're designing your primers. So during PCRs, the annealing time of primers to the DNA strand depends on the length and base of the primers. Any questions? Okay. Next, during analysis of PCR reaction, the percentage of agarose gel depends on what? The size of the DNA to be visualized, the primer concentration, the number of PCR cycles, or the type of buffer used. Jagana says it is A. Vivek also says that it is A, the size of the DNA to be visualized. Let's look at it. So, how do you analyze if your PCR has worked? We have seen how we do it previously as well. If you want to see how much DNA you have, you can load it into a agarose gel, and using UV, you can visualize it when you stain the entire gel using ethidium bromide. The same thing happens. You take your amplified product and load it onto the gel and see how bright the band is. So once the PCR cycle is completed, the amplified product is loaded into agarose gel and absorbed with ethidium bromide staining under UV source. You can include a negative control, such as just the water that was used to set up the PCR reaction to make sure that you do not have any contamination from any other DNA. And then you can also include a positive control, which lets you know that your PCR reaction has worked, and that will tell you that if any of your reagents has gone bad or not, because then the positive control will also not give you a result. If your positive control gives you a result and your test PCR does not, then it means that your template has gone bad. If both your positive and your test does not give you a result, then that means that your reagents have gone bad, and you then optimize accordingly. So, and finally, what percentage of agarose gel would you make? So normally 0.8 to a 1% gel is used to record or analyze a zero point 0.5 to 5 kb amplified DNA. But if you have a larger or a smaller size of DNA, then you can adjust the percentage of the gel accordingly. So if you would remember when we were discussing about agarose gel percentage actually determines how much cross-linking will there be between the different in the gel. So the higher the percentage of the agarose gel, the higher number of cross-linking and the slower the DNA would move through the gel. The smaller amount of cross-linking, the smaller amount of percentage of the gel, the faster that the DNA would move. So, depending on how big of your DNA fragment is, Say if you have huge DNA that you are like 10 kbs, 12 kbs, 20 kbs that you have amplified and you want to analyze, then you make a very low percentage gel so that the DNA can move and you can resolve the gel. You can figure out where it will land along these bands. So if this is a ladder, so the top bands are all 10 kbs, 8 kbs, 6 kbs, 4 kbs. So if you have a low percentage gel, then these top bands will be able to be separated very well because the DNA will move faster. And then your huge amount of DNA, you will be able to figure out, yes, this is a 8 kb band or 8.5 kb band. But if you have a small DNA, such as zero point 0.5 to 5 kb long, then you want these bands, which are lower than 3 kb, to be resolved well. And these they will resolve well at a 1% agarose gel. And then if these will resolve well, then you are able to figure out, yes, if this is 3 kb, this is 2 kb, this is 1.5 kb, then my amplified DNA of interest is 1.5 kb. So this is how you analyze the result of the PCR. So during analysis of PCR, the percentage of agarose gel depends on the size of the DNA to be visualized. Any questions? Okay. One of the major PCR disadvantages is there is no requirement for specific primers. There is a need for target DNA sequence information. The PCR is unable to amplify short DNA sequences, and the amplification process is slow. What is a major disadvantage of PCRs? Jagannath says it is B, need for target DNA amplification. No, it is the need for target DNA sequence information. Anyone else would like to take a try? Vivek says it is D, that the amplification process is slow. Okay. Do we have anyone else that can try and answer? No. Okay, so we'll look at it. So, what are the limitations of PCR? The first limitation that you have, so PCR is an incredibly powerful technique. The PCR limitations of PCR are very short, essentially, and the ones that are very obvious are going to be the first is that you need to have the target DNA sequence information. If I give you a DNA and I say just amplify this for me, you won't be able to because you need to synthesize the primers that will bind complementarily to the target sequences. So you need target DNA sequences. But once you know the DNA sequence, we just have to synthesize the primers commercially, and there are huge amount of companies that do it for a very cheap cost. Then you can just synthesize the primers. But the first step is knowing the target DNA sequence information. The next is a infidelity of DNA replication. This means that you will always have some error whenever you are amplifying. For example, TAC-Pol does not have any proof reading mechanism, which means it does not whenever it amplifies just in one go. It will not go back and check if it has put in a wrong base. So it doesn't have a 3' to 5' exonuclease activity, which means that it will keep, or there is a chance that you will have wrong bases being added. Further, tacpol also has this particular characteristic that it will always add a final. Hello, wait a second. I think I got disconnected. Mm-hmm. Okay. Can you guys hear me now? Okay. No. Yeah. Coming back. Infidelity of DNA replication. The tagpole is doesn't have any proofreading activity, does not have a 3' to 5' exonuclease activity. So you will have errors in the replication process that will happen. Further, tagpole, as I said, has this characteristic that it will always add a final A to the strand, so you will always have one A to the ends. So it is error all not error prone, but like there are errors that can creep in. But again, there are ways around it. You can not use tacpol. There are other polymerases nowadays that can replace stack polymerase, such as there is PAFU, there is fusion, and all of these polymerases are better, much better than tacpol in particular steps, such as PAFU and fusion have a very low uh, error rate, so they will keep on adding bases at a very high accuracy. And fusion especially can amplify huge amount stretches of DNA. For example, Taq polymerase is able to synthesize up to 3 kb, and after that it will not be able to synthesize. You won't be able to amplify more than that big of a DNA stretch. But using fusion polymerase, you can synthesize a lot more. You can synthesize up to 10 kb or something. So there are ways around it. Next is up to 5 kb can be easily amplified using PCR. Up to 40 kb can be amplified with some modifications. But you cannot obviously amplify something like that is 100 kilobase or bigger, and that poses a problem. But again, there are solutions. So for example, in the human genome project, we did something very interesting. We did something called shotgun sequencing, in which instead of amplifying the entire genome and sequencing it, we broke the genome down into smaller and smaller fragments that could be amplified using PCR and then sequenced it, and that made it a very easier process. So yeah, limitations of PCR. And the ones that are here, one of the major PCR disadvantages is that you need to have the target DNA information. Any questions? Okay, next. The oligonucleotides between blank is the ideal length for primers used in PCR reaction. Jagannath says it is D, 18 to 24 bases. Any other? Uh, Anyone else wants to try and take a guess? Okay, so this is something that we just discussed. Uh, So these are different reagents that you would require for polymerase chain reaction. The first is the template DNA that you want to amplify. So this can be all the way from 1 picogram of DNA to 1 nanogram of DNA. So extremely small amounts of DNA, and you can make like micrograms of DNA out of this. And this is for short templates or viral sequences. And in case of genomic DNA, you have to use a larger amount, 1 nanogram to 1 microgram. The reason being that genomic DNA is first huge, so it will weigh a lot. So, for example, in 1 nanogram of 1 kb template, there will be a lot of template copies. But in 1 nanogram of genomic DNA, there will be far fewer number of genomic DNA copies, because obviously the weight of the genomic DNA is much bigger. Secondly, genomic DNA will only have your sequence that you want to amplify. probably only two times on both the chromosomes. Yeah. So you need a larger amount of DNA in case of genome NTAs. Then next primers, you use zero point 0.1 to zero point 0.5 micromolar of each primer, forward or reverse. Magnesium chloride, it is optional, and this you put in if your PCR is not working. DNTPs, these are the four ATPs, GTP, CTP, and TTP that you actually want to polymerize into the new strand. Tag DNA polymerase, the amount varies zero point 0.5 to 2 units depending on the reaction volume. And then you add water to actually make it up to 50 microliters or 20 microliters, whatever you're setting up. And we have already seen how the different points that are important for designing primers. And 18 to 24 bases is the ideal length to ensure specificity as well as to make sure that they are short enough to quickly bind to the target DNA and not have the two target DNAs re-anneal to each other. So the oligonucleotide between blank is the ideal length for primers used in PCR reaction. It is 18 to 24 bases. Hmm. I don't seem to be missing a slide here, but it's fine. Okay, so what the next question is, primer melting temperature depends on which of the following: buffer composition, primer length, GC content of the primer sequence, or the target gene size. The primer melting temperature. Okay, so let us look at it. So we have seen what the primer designing normally entails, the primer length, the melting temperature of the primers, and the annealing temperature as well as the specificity is something that you have to keep in mind. And now primers, as we have discussed, the melting temperature you can calculate using these two formulas. The first is 4 into number of GCs plus 2 into number of A or Ts, whereas otherwise you can use this formula as well. But all of these depends on the buffer composition as well. So hybridization or annealing or melting of DNA depends on a lot of factors, majority of which are determined by the temperature, the length, the sequence, as well as the salt concentration of the buffer in which the primer is there. The reason being that we know that the primers are DNA strands, and DNA strands are negatively charged. So even though hydrogen bonds pull them together and keep them in a double helix structure or a double stranded structure, they always try to pull apart from each other because they are negatively charged and there is a repulsion. So, a very important factor for melting of primers or annealing of primers is going to be the buffer composition. So, if there are a lot of salts in the buffer that you are doing all of this in, then the negative charges of the phosphate backbones of the primers will be masked by the different positive counter ions going around. For example, say you have sodium chloride or magnesium chloride added to your PCR mix, then when the primers try to separate out from each other, magnesium will make to form a mask around the negatively charged phosphates, and this will cause the backbone to not to be able to pull. You have to put in more energy for it to be pulled apart because magnesium, the electrostatic charge and the repulsion is not there anymore. Similarly, annealing again, when you have annealing is easier when you have a higher salt concentration in the particular buffer. So this is something that you have to keep in mind. And thus, my primer melting temperature not only depends on the primer length or the sequence, but also the buffer composition. And sequence matter obviously because we know that the G and C's can hydrogen bond, they have three hydrogen bonds between each other, whereas A's and T's have two hydrogen bonds between each other. So it is much G and C's are much more difficult to separate compared to A and T rich primer. So if there are a lot of G's and C's, you have a lot of percentage of G's and C's, then it is difficult to design that primer because it will go and bind to somewhere and not try to melt at all, and that causes a lot of problems. Next, if interaction between two different primers causes a band to appear on the gel, it is called same sense primers, hairpins, repeats, or cross dimer. So we'll give you a minute to figure this one out. Okay, let us look at it. So again, the next step in primer, the this is now we're going to deal with how to optimize your PCR if it is not working. So if you do not see any band on the gel or see a lot of bands, so that could there could be a lot of reasons because of which your that appears. So the first is primers can form secondary structures. If you have longer primers that are self-complementary, so they will bind, the same primer will loop around and bind to itself when the PCR goes to the annealing temperature, and that will cause the primer to not work well because it will not bind to the stretch of DNA that you want. So the primary secondary structures arise as a result of intra or intermolecular attraction within the primer or with other primers, which eventually reduce the yield of application as availability of single-stranded primers will be limited for PCR. And these could the all of these structures could be either hairpins. In the case of hairpins, the ends of the primers actually loop around and base pair, the middle does not. The hairpins are loop structures formed by intramolecular interaction between the primer. Optimally, a 3' end hairpin with a delta G of minus 2 kilocalories per mole and an internal hairpin with a delta G of minus 3 kilocalorie per mole is tolerated generally. So essentially, this is the probability with which it will base pair with itself, and you want it to be as low as possible. And next, a dimer. This is something that we always see on our gels. If your luck is not good, so a primer dimer is a structure formed by double-stranded light structures which are formed by intermolecular interaction between two primers. So essentially, instead of the primers binding to the target sequence, the primers bind to each other. And then you only have amplification of very short primers, and that will cause your entire PCR to go. If the interaction is found between two homologous or same sense primer, it is called a self-dimer, whereas interaction is found between two different primers, it is called a cross-dimer. Optimally, a 3' self-dimer with a minus 5 kilocalorie per mole delta G and an internal self-dimer of minus 6 kilocalorie per mole is tolerated generally. So again, you do not want your primers to bind to themselves. If the primers bind to themselves, then they will not bind to the template, and then you will have a band which is very small and will be at the bottom end of your gel, and then you will have no PCR application. And you generally want self-dimerization or cross-dimerization to be as low as possible. Repeats and runs. Repeats are consecutive occurrences of dinucleotides, whereas runs are continuous stretches of single nucleotides. The maximum number of repeats and runs is 4 dinucleotides and 4 base pairs, respectively, because again, these will form secondary structures. And primer template homology. Primer should be designed in a way that there should be no homology within the template other than the target site. This will result in non-specific binding and amplification. So this is easy to design in case of plasmids, which are small, say 5 kb to 15 kb long, or 10 kb long, and you can actually go around and see, yes, my primer does not bind to anywhere else. This is extremely difficult in case of genomic DNA. Even if you run a primer blast, when you do your PCR amplification, you will see 5 different bands which are not the size that you want. That is because genomic DNA is huge and the primer cannot specifically bind anywhere it wants, and then you will have a lot of bands. But this is part and parcel of the PCR, and you will want the least amount of homology between the primer and any other places in the template. So if interaction is found between two different primers, it is called a cross dimer. Okay. Okay. Next. The number of GC content at the 3' end of the primer should not be more than 3 to prevent what? The options are primer dimer formation, GC clamp, excessive elongation, or reduced primer specificity. I will give you a minute to figure this one out. Okay, let us look at it. So, another point that you have to make sure to keep in mind during designing a primers is that the number of G's and C's in the primer should be 40 to 60% and not more than that. The reason being G's and C's are bind very strongly to each other. And so if there are, even if there are mismatches between the primer and a non-specific target location, then if the G's, there are a lot of G's and C's, they will still bind somewhere. And then that will cause non-specific elongation. And the final is prevention of a GC clamp. As GC forms a stronger bond than AT, the number of GC contents at the end of the 3' of the primer should not be more than 3. Otherwise, it will bind not specifically tightly bind to regions where GC are abundant, So your primers, instead of binding from 18 to 24 base pairs, it will just bind at the end at the three GCs that you have added, and then it can bind anywhere now, and then it will give you a lot of non-specific bases bands. Okay, and again, as I said, designing of primers, you can do a lot of smart things while designing primers. For example, say that you want to clone the sequence that you have amplified. So say you have designed a forward primer like this and a reverse primer like this. If you want to clone using restriction enzymes, then you can add different restriction enzyme sites at the ends of the DNA. So after the PCR, the primer sequence is incorporated into your uh, amplified PCR product. And now, if I want to clone it into a plasmid that has BAM-H1 and XHO1 sites, then I can restriction digest my PCR product with BAM-H1 and XHO1, and be a PCR restriction digest my plasmid with BAM-H1 and XHO1, and be able to incorporate the PCR product into my plasmid. So essentially, you can use PCRs to clone your DNA of interest into a specific plasmid with the compatible restriction sites. So this has been done a lot of times. The other newer method is using something called TA cloning, in which you use the inherent property of Taq polymerase that always adds A at the 3' end, and you design a plasmid that has a T at the 5' ends. So that due to that single base pair hydrogen bonding, you can easily clone your PCR template into your plasmid TA cloning plasmid. The other way you can use it is using blunt-ended cloning. If you use a PCR polymerase such as fusion, which produces blunt ends, and you have a blunt-ended plasmid, then you can always clone that into the plasmid. Just the efficiency is lower than TA cloning, but these are very standardized strategies to clone your gene of interest or a specific part of DNA into the plasmid of your choice. And again, different steps in a PCR reaction. A number of GC content at the 3' end of the primer should not be more than 3 to prevent a GC clap. Okay, next, real-time PCR, which is another form of PCR that is extremely common and popular, is also known as conventional PCR, reverse transcription PCR, sequencing PCR, or quantitative PCR. I will give you a moment to figure this one out and see if there are any messages. Okay, that is enough time. Let us look back at the question. Real-time PCR is also known as what? So now we are going to discuss RT-PCR or qPCR or real-time PCR, and this is a very powerful technique, even more powerful than the normal PCR, but it has a specific use case. So what is a real-time PCR? The real-time PCR combines the amplification of target DNA and its detection in a single step. It collects data while the DNA is getting amplified. So there are various techniques on how real-time PCR works, but real-time PCR combines the amplification of gene of interest with the intensity of fluorescence. This is the modern way of how we do it. The moment at which the target amplification is observed first mainly defines the reaction. The time period at which fluorescent intensity exceeds background is known as cycle threshold or CT. We'll see what this means. And as a result, a large amount of target DNA, the fluorescent signal will appear more quickly, resulting in a lower CT value. Again, we'll see how this works. The other names for RT-PCR are kinetic chain PCR or quantitative or qPCR. So how does qPCR or quantitative PCR work and why would you use it? The reason for using it is say you have a specific amount of DNA that you do not know how much is there. And that is a very, very small amount. So say you have a fragment of DNA present in a reaction mix, but it is at the picograms or nanograms level that you cannot see on the gel. So how do you determine how much DNA do you have? You use PCA, quantitative PCR method for that. What you do is that you run a simple PCR reaction with the primers for the particular gene of interest or the fragment of interest. You take your template DNA, you denature, you anneal the primers, and then you extend. But between all of this, so for example, there are various ways of doing it. One of the ways is that you have a probe. This is known as a Tachman probe. The Tachman probe actually is a small primer-like sequence that binds to somewhere upstream from the primer, and it has two different molecules bound to the ends. One is known as the fluorescent reporter, and one is called the quencher. When two of them are bound very closely to each other, then there is no fluorescence at all. But when the Taq polymerase or whatever polymerase synthesizes the strand, it actually causes this particular arrangement to completely break apart, causing the fluorescent reporter to be away from the quencher. And once the fluorescent reporter moves away, it gives a fluorescent signal. So essentially, using this technique, you are able to figure out how much piece template or how much the PCR is working. So once this is how the graph for fluorescence would look like. Initially, there would be no fluorescence, but as more and more copies of the template is getting amplified, more and more of these Tachman probes are getting disrupted. So fluorescence intensity increases in proportional to how much of the PCR is working on, how many cycles of the PCR is going on, and thus you have this very specific sigmoidal curve. And looking at the curve, you can actually back calculate how much DNA you initially had, even though you maybe have a very, very small amount of DNA. So essentially, you detect how much DNA is getting amplified each cycle using the fluorescent signal, and then you plot a graph like this, which is fluorescent versus the number of cycles. And from this, you can back calculate to how much DNA you started with. So it is quantitative in nature. So it gives you how much DNA is getting amplified at this moment, and also you are able to figure out how much DNA you started with. So quantitative, and it is called real time because you look at this amplification in real time. The PCR machine actually reads the fluorescent signal at the end of each cycle. So each cycle you know how much DNA you exactly have. So this is called quantitative or real time PCR for this reason. And it is used to quantitatively measure how much DNA you have at this moment, at this cycle, also back calculate how much you started with. And this is what the amplification curves normally look like. These are different primer pairs that they are being detected, and this is the fluorescent signal that is being detected. And then you cut the threshold off at a particular level, which makes sure that you do not detect the background fluorescence, whatever the sample should have. So once the threshold is crossed, only then You figure out that yes, my DNA is being amplified. And this is the amount of DNA that I have. We back calculate and figure out how much is started with. So in real-time PCR, the time at which fluorescence intensity exceeds background fluorescence is called cycle threshold. No, 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 no. There is okay. Okay. The next question, which is in real-time PCR or qPCR, you have just seen the time at which fluorescence intensity increases the background intensity is known as amplification threshold, cycle threshold, fluorescence threshold, or reaction threshold. You can try and figure this out. ওখানে দাঁড়িয়ে আছি মিউট করা আছে Hello, 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 hello. Okay, it seems I was disconnected. Coming back. Okay, the question was in real-time PCR, the time at which fluorescence intensity exceeds fluorescent background is called what? Amplification threshold, cycle threshold, fluorescence threshold, or reaction threshold. So this is something that we just saw. In real-time PCR, this is the curve that you would expect the qPCR machine to give it to you. So a qPCR machine is upgraded form essentially of a thermal cycler in which you can also detect the fluorescence intensity at different cycle intervals. So what you detect essentially is the cycle threshold at which at the baseline fluorescence is exceeded by the signal that you receive as fluorescence. As we just discussed, with different techniques also. If you use different cyber green or Tachman probe technique to do the real-time PCR, the fluorescence intensity should increase exponentially with time as more and more DNA is getting amplified. So you set a correct CT value or a threshold value at which this threshold is exceeded. And this CT value is what is used to calculate the amount of DNA that you started with or the amount of DNA that you have currently. Anything below that you will imagine is not being as not being amplified correctly. So this is what you would say as not detected. Anything below the threshold, anything above A was detected. So it is the cycle threshold. Next, one of the main advantages of real-time PCR is blank. First, it requires a large amount of template DNA. It gives quantitative measurements of the DNA RNA sample, cannot analyze multiple samples simultaneously, and it is less sensitive than conventional PCR. I will give you a moment to figure this one out. Okay, so one of the major advantages of real-time PCR is, so let us look at how exact different aspects of PCR, how is it different from real-time or quantitative PCR, the real-time and fundamental PCR. The first is the quantitative analysis. Real-time PCR allows you to quantitatively measure the amount of DNA being amplified at every step because you will have the fluorescence signal coming from the Tachman probes, and it gives you quantitative provides quality. Fundamental PCR just says that yes, you had DNA, you have amplified it, this is the amount. But when you look at the band after 35 cycles or 39 cycles, you will not be able to figure out how much you started with, and even the final amount is not very, you can quantify it, back calculate it very well to the amount that you started. The speed, the real-time PCR provides results in real time. So you will be able to just look at the results and see, yes, this is the amount of DNA I have at this moment. And fundamental PCR actually needs you to load the PCR onto a gel and then figure out the intensity and whatever, if your PCR has worked, if it was there or not. Sensitivity, the real-time PCR is much more sensitive because you can directly quantitate the amount of DNA from the fluorescence and can detect low numbers of DNA, target DNA due to continuous monitoring of the sample. Fundamental PCR is less sensitive. It is has a reduced risk of contamination. The closed tube system of real-time PCR reduces the contamination risks due to minimal post-PCR handling. So there is no mixing of the dye, mixing of water, the inactivation, loading onto the gel. None of that is happening because you detect it as the PCR is working. So it is there is a reduced contamination risk. Data accuracy, again, due to quantifiable fluorescence signals, you can have very high accuracy of the amount of DNA that you started with and quantify the amount that it increased exponentially. It is high throughput. So normally, real-time PCR or qPCR, when you set it up, you set it up into a 384 well plate, and you can do a lot of reactions at the same time. You can probe a lot of different uh, lot of the you can probe for a lot of the different DNA segments that you are amplifying. So from the same DNA that you start with, so each of the wells will have a different primer pair that will amplify different segments of the DNA, and then you can detect every one of them individually. And we have seen this happen in case of COVID testing. So obviously, RT-PCRs became a household term during COVID when COVID, RT-PCR was used as the test to figure out if someone has COVID or not, because you actually amplified real time the amount of different genes that different RNAs that our COVID virus has. So if you see any signal after 35 cycles, you would be able to say, yes, this person has COVID because they have the different amounts of the foreign COVID viral genes that shouldn't be there in a for negative control sample. And that is why how it was used as a diagnostic tool. And multiplexing, again, you can detect different samples at the same time because you set it up into a 384 well plate. So you can have different genes that you're probing for the same time or different patient samples that you're probing for the same primer pair for COVID at the same time. So multiplexing is also a very major advantage of qPCR over fundamental PCR. Next, Uh, what are the requirements for real-time PCR? The first one being you have a thermal cycler that can actually detect fluorescence, so it is known as a qPCR machine. Next, you have a DNA or RNA template. So DNA, as we have been discussing, you amplify the DNA and look at how exponentially it gets amplified over time, and that gives you the real-time quantitative measurement. But you can also start with RNA, and this is a much important use of real-time PCR. So real-time PCR, anyone when they say RT-PCR, they actually do not mean real-time PCR. They actually mean reverse transcription PCR or QRT-PCR, quantitative reverse transcription PCR, which we will see. And using that, you can figure out how much RNA does your sample have of a particular gene. Next, you have to have primers that you will have enzymes which are DNA polymerase and reverse transcriptase in case of reverse transcription PCR. You have to have a fluorescent dye or probe reporter such as CyberGreen or TACMAN, a real-time detection system which will be in the qPCR machine, and PCR tubes and plates. So, as I was saying, the much better use for real-time PCR is actually in reverse transcriptase PCR. So, this is a problem that happens because of the naming scheme. Even though real-time PCR is called RT-PCR, there is another PCR called reverse transcription PCR. In this, you actually do not. What you do is that you want to figure out how much of a particular RNA is present in your sample. So this is what was used majorly during COVID testing. You want to figure out how many mRNAs or how many RNA copies of a particular COVID gene is present in your patient sample. So the first step for a reverse transcriptase PCR is actually making a cDNA copy. Reverse transcription, as you would remember from our discussion on central dogma, is a RNA-dependent DNA polymerase. So you take a RNA and you make a DNA copy complementary to that RNA. And that complementary copy of DNA is called cDNA or complementary DNA. And that cDNA is what you use to do your real-time quantitative PCR. So the entire technique is known as quantitative reverse transcription PCR or QRT-PCR, but also known sometimes as RT-PCR. So whenever someone says RT-PCR, almost always they are talking about application of RNA to conversion of RNA to cDNA and calculating the cDNA because using real-time PCR, you're able to figure out how much the DNA you started with, and we know that using reverse transcription, you would be able to convert one mRNA copy into a cDNA. So the amount of RNA you have is equal to the cDNA you have. cDNA you can calculate using the real-time PCR. So essentially, you are able to back calculate how many copies of a particular RNA of interest your sample has. And this is what was used during COVID testing. You figure out the amount of COVID specific RNAs that your patient sample has using reverse transcription PCR or QRT PCR, quantitative reverse transcription PCR. So RT-PCR, RT-qPCR, all of these are actually the same thing. So whenever someone says RT-PCR, they're probably talking about reverse transcription PCR rather than real-time PCR because the small amount of DNA copies that you actually want to figure out using RT-PCR are most of the time coming from this cDNA synthesis because the number of RNA copies is just very, very small and you will not be able to figure out that using a gel. So the main use of real-time PCR is using it in reverse transcription PCR or QRT PCR. How does reverse transcription PCR work? So you have a single-stranded RNA template such as COVID viral gene. You use a reverse transcription step to make a RNA DNA hybrid. And the new DNA that is made is known as the cDNA. This then you use an enzyme called RNase H, which disrupts and destroys any bound RNA DNA hybrids and just specifically disrupts the RNA. So you now have a single-stranded DNA template, and then you actually run a PCR, and this gives you a double-stranded DNA template, which is your cDNA. So essentially, you make cDNA from a double-stranded cDNA template from a single-stranded RNA template, and using this double-stranded DNA template, you actually set up your RT-PCR, and you are able to figure out how much dsDNA cDNA you started with, which correspond to how much RNA of this particular gene the patient sample had. So the steps that are involved in this is sample preparation, which you prepare the DNA or RNA sample, primer and probe design, reaction setup, amplification cycles, fluorescence monitoring, threshold detection, and then data analysis. So this is all that is. So this is very similar to the steps that we are doing in normal PCR. Just the fluorescence monitoring step is extra. So one of the main advantages of real-time PCR is it gives quantitative measurements of DNA or RNA sample. Next, Tachman probe for real-time PCR employs what method? An RNA-specific RNA primer, a sequence-specific fluorescence probe, a cybergreen dye, or a DNA binding protein. I'll give you a moment to figure this one out. Okay, so let us look at it. So we're going to discuss the different principles of real-time PCR. The principle of real-time PCR is to quantitatively measure the amount of specific DNA or RNA target in a sample by continuously monitoring the amplification process. And this technique leverages the natural ability of DNA to produce fluorescent signal when it is amplified. So we'll see how that works. The first method that was previously used and still used, but is getting removed mostly because we have a more modern technique is called cyber green dye method. So in this method, as fluorescent dye such as cyber green is added to the PCR reaction mixture, cyber green will bind to any double-stranded DNA that is generated during the PCR process and not to any single-stranded template that is going around. So as DNA target amplifies, more double-stranded DNA is produced, leading to an increase in fluorescence, and this is directly proportional to the amount of target DNA that is getting amplified. However, it is important to design specific primers to ensure that the fluorescent signal corresponds to the specific DNA that you are amplifying, because this technique is not sequence specific. So any double-stranded DNA gets amplified, you will see a signal, but doesn't mean your double-stranded DNA is getting amplified. But essentially, whenever polymerization happens and you have a double-stranded DNA, the amount of signal essentially increases exponentially because the cybergreen will start giving you fluorescence by only being incorporated into double-stranded DNA. The other one, which is the much more used technique, is called Tachman probe method. This method employs a specific sequence specific employs a sequence specific fluorescence probes such as the Tachman probe that is designed to bind to target DNA but upstream of the primer. When the probe binds to the target DNA during synthesis, the DNA polymerase cleaves the probe, releasing the fluorescence reporter. The increase in fluorescence is directly linked to the amplification of the target sequence. And this is highly specific because only probes that were bound to the single-stranded DNA template and was removed or cleaved off due to polymerization will give you signal. The real-time PCR instrument sets a predefined fluorescence threshold, and the cycle at which the fluorescence signal surpasses this threshold is called the CT value, cycle threshold, or the quantification cycle value. The CT value is inversely proportional to the initial amount of target DNA or RNA. So the lower CT value indicates higher initial concentration of target sample, which is obvious because If you have more amount of DNA or RNA template that you started with, then the CT value will be crossed much faster. So lower CT value means you had much more DNA to start with, and then you calculate the entire thing using different softwares. So this is the technique that I was talking about earlier. So this is a TACMAN probe, which has a quencher and a fluorescence reporter, and a part that specifically binds downstream to the target sequence of interest. Once the polymerization happens, this in the 5' to 3' direction, anything that is bound gets completely chopped off. Because of this chopping off, the fluorescent reporter is separated from the quencher, and the quencher normally prevents any fluorescence. Once they are separated, the fluorescent signal now can be visualized. So more and more amplification of the template occurs, more amount of signal will be received, and that is why how you will calculate quantitatively be able to figure out whether PCR, how much DNA you have in the PCR reaction. So TACMAN probe, real-time PCR employs a sequence-specific fluorescence probe. Next, in real-time PCR, the plateau phase represents the point where the majority of target DNA has been amplified, the PCR reaction starts to fail, no amplification occurs, or the primers are exhausted. I'll give you a moment to figure this one out. Okay, so we are now going to talk about the analysis of RT-PCR. So in real-time PCR, the most common graph obtained is the amplification plot that you can see right here, which is fluorescence against the number of cycles, which visually represents the accumulation of PCR products over the course of the reaction. The x-axis is obviously the PCR cycles, and it involves all the three: denaturation, annealing, and extension. That is one side. Y-axis determines the fluorescence signal that is detected, and the amplification curve, which is each sample or target gene, is represented by a specific curve on these graphs. And these curves show how the fluorescence signal increases over the cycles. A steeper slope indicates a higher amount of target DNA. The amplification curve has three phases. The initial phase, which is here, it represents the early phase of the PCR reaction. There is not enough amplification of DNA to produce a significant increase in fluorescence, and the cycle starts with denaturation of double-stranded DNA to separate single-stranded DNA. So in this phase, even though the amount of DNA is increasing, the increase is small enough that it is not detected. And the next is the exponential phase, which is this phase, and in this phase, the fluorescence signal increases with each cycle exponentially, and it forms a well-defined sigmoidal curve which indicates the PCR reaction is proceeding efficiently. During this phase, the CT value is typically at the middle of the phase, which is used to determine the quantify the amount of initial DNA. The slope is steep if the slope is steep, which tells the efficiency of the reaction, while the linearity of the slope represents the doubling of the amount of DNA. And finally, we have the plateau phase in which the curve just stops. And this phase is where majority of the target DNA has been amplified, and the curve loses its linearity and hence is not suitable for precise DNA quantification. That is why you use the threshold to quantify in the exponential region and not the plateau. The threshold line is what is this one is set above the baseline fluorescence, and it is the cycle at which each curve crosses the threshold is known as the CT value or the CQ value. The lower the CQ value, the higher the amount of initial target DNA, as we just discussed. As it is known that CT value is inversely proportional to the initial concentration of DNA. If the CT value of one sample is lower, the amount of initial DNA concentration was much higher for that particular sample. So essentially, if this is the CT value, so on the x-axis we have the cycles. Whenever the curve crosses the threshold, you stop at that. That is your CT value for this particular DNA sample, and you would say that yes, this CT value is lower than the next one. So this particular DNA was much higher in abundance than the right one. So in real-time PCR, the plateau phase represents the point where majority of the target DNA has been amplified. Next, in exponential phase of the real-time PCR, blank with each cycle in an ideal condition. The reaction rate decreases, the fluorescence signal stabilizes, the DNA template is exhausted, or the fluorescence signal increases with each cycle. I will again give you a minute to figure this one out. Okay, so let us look at it. Again, the same particular analysis. So as we have discussed, the initial phase is the early phase of the PCR reaction, and not enough signal is observed in this phase because the amplification has just started. The amount is too low for any viable signal to be observed. The next is the exponential phase, in which the fluorescence signal increases as we see with each cycle, and it forms a sigmoidal curve, which indicates that the PCR is proceeding efficiently. So a curve like this, and this is the exponential region in which with each cycle the amount of DNA doubles, and thus the fluorescence increases with each cycle in the exponential phase. In the exponential phase of the real-time PCR, with each cycle, the fluorescence signal increases. Okay, so that is all I had for you today. You can find the recorded lectures at my YouTube channel, which is with my name in the molecular biology playlist. If you have any questions, you can drop them in the NPTEL forum or in the comments of the YouTube section video. And if there are any queries, please let me know. And thank you for joining in today. I will see you next week. Thank you.